with HPV, because as we know, HPV can be transmitted through the mouth. Therefore, it's a really big risk factor for turning into head and neck squamous cell carcinoma.
Yes. As you may heard about HPV before, this is like the very common sexually transmitted virus, which can cause a lot of, type of diseases in adult. And in this specific case, HPV positive patient or people can have like really high chance of having the cancer in the throat, which is the head and neck squamous cell carcinoma. Yeah. Okay. So yeah, here is the one thing that we want to explore for specific in this cancer, which is like a process called epithelial mesenchymal transition, or EMT. So this is a very specific process that occur in head and neck squamous cell carcinoma, in which the epithelial cell will gradually convert or transform into mesenchymal. Yeah.
Yeah, so when we're exploring cancer, one of the processes which we want to research more into is the process of metastasis. And so one of the ways in which this happens is through epithelial mesenchymal transition, or EMT. This is where epithelial cells lose their cell adhesion and gain enhanced migration and invasiveness. So in this diagram right here, you can see how the epithelial cells are adhering tightly to each other with these tight junctions. Then as they transition into mesenchymal cells, they lose this tight junction, making them more prone to migrate to other parts of the body. And so our research question that we're interested in exploring today is, what are the molecular mechanisms of EMT within head and neck squamous cell carcinoma? And how does this contribute to the metastasis or the spread of the tumor cells?
Yeah, yes, sure. Okay. Uh, okay. So now we dive directly into what we have found with the head and neck squamous cell carcinoma. And please, along the way, please feel free if you have any questions or anything that you would like us to explain further, please just type into the chat or the Q&A box so that we can, talk back and forth right away in the section. Okay. Now we go to the first part, which is to explore the gene expression patterns across the Talk to Data beta version.
Okay, so here it's known in previous literature that the loss of CDH1 expression is involved in tumor cell invasion and metastasis. And we want to see if this pattern upholds in head and neck squamous cell carcinoma as well. So when I filter for epithelial cells within our digestive system and I compare our head and neck cancer versus our normal, we can see a slight loss in CDH1 expression in our cancer condition compared to our normal. And the expression level that we're looking at here is the average rank, meaning the higher the rank is of that gene within that cell, that means that cell is expressing that gene very highly. And over here in this detailed box, it shows you the amount of studies and the amount of samples that were pulled together in order to query this expression level of this gene. And you can see that we pulled the analysis from four studies across 23 samples. And of those cells, about 55% of them express CDH1 at this lower level right there.
Yeah. I think like it's a little bit, it's quite clear that like the expression level of CDH1 in head and neck epithelial cell, head and neck cancer epithelial cell is a little bit lower in terms of expression level. However, in terms of the coverage of this gene, I believe is quite similar. So the proportion of cells, like of either normal epithelial cells or cancerous epithelial cells expressing this gene, is quite similar. In here, I believe it's about 50% of the cells expressing CDH1. However, the expression level reduced a little bit in here.
That's right.
Yeah. Okay. And after that, because we found CDH1 because CDH1 is a very popular gene in cancer, which associate with like the metastasis and also the invasion of cancerous cells. So that our next question would be, what other genes are like co-expressed with CDH1 in HNSCC epithelial cells, so that we can find like a bigger gene panel related to this cancerous epithelial cell.
Mhm.
Okay.
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 So then on Talk to Data, we can perform a co-expression analysis. So if I query for CDH1, it will return the top 10 genes that are co-expressed with CDH1. And we can see here that this top gene right here, MAL2, is highly co-expressed with CDH1. This gene is related to intracellular trafficking. And when we do a literature search on MAL2, we also find that there's high expression of MAL2 also detected in breast cancer tissues and cells. And so with this result, the fact that CDH1 is co-expressed with MAL2, we want to see, well, do these epithelial cells that co-express these two genes, do they display some type of EMT trait?
Yes, sure. Okay. In here, basically, we found CDH1 and MAR2 being the two most co-expressed in the head and neck cancer epithelial cells. And then
Mhm.
we use those genes to further define the epithelial cell subset. And there's one thing that I want to mention here, that the co-expression map right here, if you already used to the current Talk data, you might knew that in the current Talk data, we can find the co-expression level across the whole database. However, in this beta version, we are now be able to find the co-expression level in a very specific condition. In this case, this is the co-expression levels in epithelial cells of head and neck squamous cell carcinoma.
Right, that's right.
Yeah. So yeah, so here just what we just mentioned, that we actually, based on the co-expression results that we found, we actually create a subset of epithelial cells according to CDH1 and MAR2 expression, so that we expect that like, does this subset present like a very clear characteristics of EMT, since they are like express genes corresponding to the cancer or like the metastasis process.
Mhm.
Yeah.
Yeah. So here in this analysis, again, just to get an overview of the expression of CDH1 and MAL2 in our cancer, CDH1 is a little bit lowly expressed in our cancer compared to normal. However, MAL2 is more highly expressed in our cancer compared to normal. And so given these results, we want to subset these epithelial cells that express a low level of CDH1 and also a high level of MAL2.
Yes. This
Mhm.
is actually quite interesting for the fact that we found these two genes are like highly co-expressed with each other. However, the pattern of these genes are like in reverse of each other. While CDH1 is decrease in the disease, and MAR2 is increase in the disease. So that's one thing we found quite interesting to know. Like,
Mhm.
regardless of the co-expression level, ones can be high and ones can be low in the disease.
Mhm.
Okay. So here, yes, this is the question that we want to answer after we create the subset. So, do CDH1 low and MAL2 high epithelial cells display a clear EMT characteristics or not?
Mhm.
Okay.
Okay, so before we get to the results, I can demonstrate how I do that within Talk the Data.
Yeah, sure.
Yeah. So here, I'm going to start adding the filters to my tree. So I want to look at the digestive system,
Mhm.
and I'm interested in only epithelial cells.
Okay. So here,
And
what Julie is doing is she's creating the subset of cells.
the
Okay.
head and neck squamous cell carcinoma, and also our normal condition.
Okay.
Okay, and so adding all these filters kind of narrows down the database to studies that are only related to the filters that I added. And then in order to subset these epithelial cells, I can go into the expression rank filter here and add genes according to their expression level. So I'm looking for CDH1 low.
Okay.
For example, if I restrict this to a lower level, CDH1, click
Okay.
apply. And then I'm also interested in MAL2
Okay. So this is
and a
actually
high.
the distribution of the MAR2 or like the gene expression. So this is the distribution across the whole database.
Mhm. Then going over here to the left-hand side, I want to find what types of marker genes are upregulated. So
Okay.
as you can see, all the filters are still applied here. And I want to select the condition and compare the two conditions to see what kind of marker genes are different between the two.
Mm-hmm.
So then
In
when I
here.
run the analysis, open up the results page, it
Mm.
will return the top 10 marker genes that are differentially upregulated in our normal. And then down here, it returns the top 10 marker genes that are differentially upregulated in our cancer. So
Mm-hmm.
going back to the slides.
Yes. Okay.
Okay, here are the results page. So I found that in this subset of epithelial cells, it displayed these marker genes that were related to cell adhesion. So for example, Gage2A is expressed in many tumors. It's also a prognostic marker in hepatocellular cancer. FAM46B can inhibit cell proliferation and cell cycle progression in prostate cancer. And then interestingly, PVRL4 is involved in cell adhesion and is found in breast cancer tumors. And we know that cell adhesion is an important trait in EMT. So this leads me to believe that perhaps this set of epithelial cells could be cancerous and may display some sort of trait that is involved in metastasis.
Mm. Yeah, sure. So yeah, I think like these the upregulation of these genes quite like partly prove that the specific subset of cell actually does represent a very specific EMT trait in which like the cell adhesion decrease and also the cell progression, the cell cycle progression actually like also increase, that kind of thing. Yeah.
Mhm. Okay.
Okay.
And so, given that, I wanted to dive a little bit deeper into what other genes are upregulated in our cancer. So I performed differential gene expression analysis, and it revealed even more upregulated genes related to EMT. For
Mm.
example, we found that SNAI2 and HIF1A downregulate E-cadherin during EMT. So you can see that here displayed in the volcano plot. And then we also have a heat map view where it shows us within each sample what the expression level of these genes are in each of our conditions.
Sure. Julie, in here, I think that it would be nice if we can explain for all of our friend here, is like, why do we perform the marker gene and then we perform the DGE? So what is the reason behind that kind of analysis experimental design?
Okay, so I think for marker gene, it's a good way to get an overview or like a quick view of what kind of genes are upregulated between two different conditions. However, when we perform differential gene expression analysis, it gives us a much wider panel of genes for us to look through, as well as the statistics involved in the differential gene expression, such as the log2 fold change, and you can see with each individual sample here what the exact expression is.
Yeah, sure. So like basically the DGE serves as like a bigger picture. We can view the whole expression profile. While for the macro gene, what we really focus on is like the genes that are only expressed in one group, but not the other. So the coverage of one group might be nearly zero, whether for the other group, it could be really high coverage. Yeah, so that's show a really difference. Oh, in here, before we move on, we have one comment and one question from Perimo. First of all, let me here like answer here. So first of all, thank you, Perimo, for your compliment. Perimo sent to us like this is a good analysis, and she she asked for like can we do a similar analysis where one gene is expressed while the second one is not in the cell type or condition, which I believe we can do. So Julie, I think we can perform like a little
Yeah,
demo for one is expressed where the another is not expressed in one subset.
yeah. So
Okay.
going back here to the expression ring filter, you can see how it has this and here. So it's saying that we want to query for the expression of CDH1 and MAL2. However, if we get rid of it, another way we can add this in is by adding it next to the CDH1.
Mm-hmm.
That way you can query for CDH1 or MAL2. Another way you can change the filter is by clicking on it. And here's where you can add that not filter. So this means that in epithelial cells, in our digestive system tissue, in our different conditions, we don't want any expression of CDH1.
Yeah, sure. Basically, in here for the expression rank filter, you can filter out the cells according to the gene expression profile. So the condition can be like one gene and another gene and one gene or another gene. And in this specific case, it can be one gene or not another gene. And besides that, you can do like what Julie just did. Is like she's not actually completely eliminate the gene from the subset, but she just select the cells that express low level or high level of a gene. So that could be multiple ways for the cell selection.
Mm-hmm.
Okay. Yeah. So yes. Okay. Thank you, Paramo. Now we can move on with the next part of the analysis. Okay, so this is kind of like the summary of the major genes that we have found along our analysis. As you can see here, we got quite some genes. Like the first two, the CDH1 and the MAR2, we found during the gene query and also based on the literature and the co-expression analysis. And then the other genes like the GH2, the FAM46B, the PVIR4, or like the SNI2 or high the HIF1A, other genes that we found along the marker genes analysis and the DGEs, which like all increase in the HNSCC, and mainly they are involved in the in cell adhesion and also the cell cycle progression. Basically, these genes again prove that the epithelial cells in head and neck squamous cell carcinoma actually does show the characteristic of EMT process.
Mm-hmm.
Okay, yeah. Okay, so like that's about the exploration of single cell RNA-seq data using the total data beta version. And after we already summary some genes that we found, we also want to highlight some features of total data beta version, like we can query some genes, we can perform multiple kind of analysis to find like important genes for specific conditions. And then what we also want to focus here is that you can be able to perform the analysis of millions of cells in like a few seconds for one analysis, which is, I believe is quite impressive to us. And then, coming from here, we will then move on to the next part of the analysis, which we will include the spatial context into our experiment. However, before we move on, we would like to ask you a little bit question about like So based on these results, what analysis would like to perform further so that we can continue to perform more analysis to show you in the upcoming sessions, or we can include these analysis into our tune. And then yes, just please spend a little bit of time and answer the survey that showing on the screen right now, so that we can improve our analysis and then our tune at the same time.
Yeah, so basically, given these results that we found, what kind of research questions would you ask further from here? And with those research questions, could we be able to answer them with talk to data?
In here, we actually got some analysis that we are suggesting, like the pathway enrichment analysis, the ligand receptor analysis, the trajectory of the pseudotime analysis, and any others analysis that you would like. Please let us know. Yeah, thank you. Okay. Okay, so I think that's it. Now we can move on to the next part, which is, to include the spatial data into our experiment. Okay.
Okay, so the spatial data that we took this from is from this study, snail-regulated exosomal microRNA 21 suppresses NLRP3 inflammasome activity to enhance cisplatin resistance. So in this data set, they have two different types of samples. One where they took a sample from the invasive front of the tumor, meaning this section right here is the tumor, and it's interacting with these host cells here. And then in the second sort of sample, they're taking it from the inner site of the tumor. So this entire tissue that we're looking at here is the tumor site and the tumor cells.
Just for your information, the host cells in here, according to the authors, are like the immune host cells. Yeah. Okay.
Okay, so in this first analysis, I just wanted to confirm whether or not we can see the co-expression of CDH1 and MAL2 in our tissue samples. So on the left-hand side, when I perform cell type prediction on our samples, this is Visium data. It was able to predict that these cells were abnormal cells or otherwise cancerous cells. And then on the right-hand side right here, is each of these spots is co-expressing CDH1 and MAL2. So there is a significant overlap of our abnormal cells and our co-expression of our two genes.
So these abnormal cells can I think it's pretty clear that these can be cancerous cells. That's right.
Yeah, that's right.
Yeah, and in here, as I noticed that in this inner side of the cancer, beside the abnormal cells, it seems that we also have other cell types, as well as you can see that some regions are not highlighted here. So we suspect that these cells may be the immune cells that are really invaded into the inner side of the tumor. Yeah.
Mm-hmm. Yeah, so from this analysis, I want to see, okay, well, are there any pathway differences between different areas of the tumor? And to look at the pathways, I can perform region segmentation. And so what region segmentation does is that it kind of classifies or it separates different regions of our tissues based on both the expression data and also the spatial context of the tissues to try to group together similar regions and then find pathways that are enriched in those regions. And so looking at the invasive front of our tissue, it was able to segment the tumor region separately from the host region here, which is in the pink. And then when I performed a pathway enrichment of this purple site, the tumor site, I found that it was enriched in the SYNCRAS dependency signature pathway, which is actually an EMT-related pathway. So it's nice to see that there is some confirmation that this tumor site, the invasive front of the tumor site, is related to EMT or related to metastasis.
Yeah, sure.
And then in contrast to that, On the right-hand side, we're looking at the inner site of the tissue. It's segmented into two different regions. And in this purple region right here, we found an enrichment in this GCM TPT1 pathway, which is a pathway related to cell growth and proliferation. So these pathways are kind of promoting the growth of the tumor cells.
Yes. In here, for the invasive front image, I think it's quite impressive that the tool can very clearly separate the pink region, which is the immune host cells, uh, to the actual like tumor region, which I can see like the purple one, based on the like the cell type prediction result, and also the pathway enrichment here seems to be the cancerous cells, and then the green regions, which might be the like based on the cell type prediction and also the pathway enrichment result might be the immune cells that are invaded into the tumor region. Yeah.
Mhm.
And the pattern is quite similar to the inner side of the tumor, like the image on the right here. We also have the cancerous cells and then the immune cells surrounded that. Yeah.
Mhm.
Yeah. Yeah. So I think that's also everything that we have found today. So I think we've gone a little bit, we have like from the single cell analytic data to spatial data, where we kind of like further confirm our findings in the single cell, and then we also find more more like evidence to the pathway enrichment analysis.
Yeah,
Yeah.
so to summarize, we found that CDH1 is lowly expressed across multiple cancers, including head and neck squamous cell carcinoma. And we found that MAL2 is commonly co-expressed with CDH1 in epithelial cells. And so when we subset the epithelial cells that express low level of CDH1 and a high level of MAL1, they displayed characteristics of EMT through genes involved in cell adhesion and E-cadherin suppression. And then lastly, in the last part, where we bring in the spatial data, we found that the site of metastasis, that inner front of the tumor, is upregulated in EMT-related pathways as well.
Sure. In here, we have received one question about the region segmentation,
Mhm.
which is like how the segment is defined and whether it's from the cell type specific markers.
So it doesn't only take into account the cell expression, but it takes into account the spatial context. So it uses what's called a Morin's I-score. So each region is assigned a specific score. So depending on that score, it segments into similar regions, kind of how clustering works.
Yeah.
How in the vein clustering, it takes into account similar gene expression profiles. However, with region segmentation, it also adds in that spatial context as well.
Yes, I should consider this region segmentation as quite similar to the Leuven clustering, like the overall expression profiles instead of like cell type specific markers, because actually we don't define any cell type here. We define the regions. And
Mhm.
and then for this tool, we also include, the spatial context, which is the Morena score, as what Julie just mentioned. And I believe, if you want to find more about the Morena and also how we define the region segmentation, we can put in the link to our document here so that people can go in and read more about how we actually define the regions here. Okay. And in here, we got another question about where can I find recordings for this and the previous club presentations? Okay, actually, all the recordings for this session and the previous sessions are all can be found on our YouTube channel. So here now, my teammate will put on a link to our YouTube channel, so you can rewatch all of the sessions in the SCC Club. And once you already got to our YouTube channel, you can watch other, guideline or introduction videos about other platforms and other features from bioturing as well. So I think it's a very good way to get to know with bioturing. Yeah. Okay, so after the summary, we will give a little bit of time for the Q&A section, and also it would be very, very helpful if that you guys can fill in the survey here, because in here we have put down some questions regarding to either you have the need to, or like some filters or criteria that you would like to have to further subset the cell populations, because what we have done today is we used ontology, different conditions and cell types, and then the gene expression level to define a subset. However, please let us know if you have any other ways that you would want to use to filter your cells. And then, since we just work with the single cell and the spatial, we would also want to ask you, like, what other types of data would you be interested in searching for? Like maybe something we don't have yet, and we will include it in our database, like the lipidomics or the metabolomics. Yeah. Yeah. So please fill in the survey. And thank you so much for your time and your comment here. We're still waiting for more questions. So please let us know if you have any question. Okay. Okay, so I think that's it for today. Thank you so much for joining us today, for giving us question and for your time. And yes, see you next week for another session. Yeah, bye.
Thank you, everyone.
Thank you, bye.